I would like to thank the organizers for inviting me here. It's really also a great program and a great conference. Yeah, indeed, I will talk about an approach to surface or membrane binding proteins that will involve concepts of surface phase separation, pre-wetting, but also classical wetting. So let me quickly have at least one introductory slide. We'll go on the other side. Obviously, we have seen lots of cases where biomolecular condensates wet surfaces, in vitro and in vivo, and here I just cherry pick a few examples. For example, in vitro contexts, for example, in the research of cruacivates, we see that lipid vesicles get deformed by cruacivates and vice versa. And we have also other examples more in vivo, for example, drug carrying liposomes get wetted by RNA protein droplets. And for the in vivo context nowadays, you can cite, of course, a lot of references, but let me pick this seminal study from Cliff, Tony, and Frank with the P granules and the C elegans. And you see already here, that's indicated the cell nucleus, and here you see the P granules, they're already wet the nucleus. And this movie is buried in the SI, and it's also cited because it indicates all these beautiful properties of biomolecular condensates, that dynamic, wet, relaxed circle shape, etc. But obviously it also shows that they undergo wetting. And what do we know from classical equilibrium wetting? As a short recap, what we know is that when condensates wet a surface, we have a triple line where all three phases coexist. And at this triple line, we know that at least at equilibrium, the law of Young and Dupré exactly gives us a relationship between the respective surface tensions and the contact angle or triple angle. And this actually emerges at equilibrium from a force balance, or when you calculate the work and you displace the contact line, that the work vanishes at that point. What is very interesting about the shape, at least when there is no external potential, such as, for example, gravity, or in other words, when the size of the condensate is below the respective length scale, for example, for gravity, it's the capillary length scale, then droplets actually have, when they're at equilibrium, a spherical cap type of shape. So if you would extend this would become a perfect circle, or in 3D, it would exactly match with a sphere. Okay. Obviously, we have these two phenomena, partial wetting and complete wetting. And here, that's the case of partial wetting. Complete wetting would be simply you have a flat layer of this blue droplet. There are, of course, many possibilities to get deviations from a circle shape. Here also, I'll quickly outline here the reference. In case someone wants to read classical literature on this, I can highly recommend this book. And if you want to see deviations, there are many ways. And one recent way we actually worked on, also together with Frank, is on chemically active wetting. So there we looked at a binding process that was maintained out of equilibrium. And then even though this process is pretty much localized here on the triple line, you can get significant distortion from a spherical cap shape. However, when you think about wetting on a biological surface, you have to think about the membrane and a biological membrane. And a biological membrane is not as simple as, of course, a solid surface. It has lots of features. Here I highlight only a few. First of all, membrane is composed of many components, lipids, actually not just one lipid, actually a few hundreds of different lipids, and of course proteins that are embedded in this membrane. And the interesting property first, because it's a liquid, both the lipids as well as the proteins that can diffuse. That's one key property. Moreover, some of the proteins, they're called receptors, they can attract other kind of proteins with rather specific interactions through binding. So this can actually lead to a recruitment of other proteins, building kind of a small layer on top of the membrane. So these are all properties classical surfaces not necessarily have. And moreover, not always the case, but of course, binding can be maintained out of equilibrium, either by maintained out of equilibrium by some few molecule. A good example is the kinase phosphatase cycle. And we call these systems, and I think we have alluded to this already a few times, chemically active. However, there's a problem. When you look at the class of membrane-binding proteins, they are often of rather small concentrations. Usually the range is a rough range, but from 1 to roughly 100 nanomolar. And when you look at their corresponding saturation concentrations, at least when they are known, this is far below So actually, from that message, would be a bad message, so my talk would be over now. There are no droplets, definitely not in the bulk, because they're extremely subsaturated. So the question is, can they still maybe organize a surface? And when you just plot a simple Huggins model of that kind, but including a surface, a phase diagram would look like this, just a simple binary flow Huggins model. So this is the interaction strength, so the more you go up here, the more adhesive you are, and this is the protein interaction. And this is now logarithmic axis because I want to extrapolate to very small concentrations. That's why, but the shape is a bit unusual. And what you discover here is, of course, a classical binodal. Within you have phase separation, and then you have two zones. One is complete wetting here, and one is partial wetting, that I've already shown to you. And here's the critical point for phase separation. And there's something else, another kind of phase transitions I want to quickly summarize. I have to zoom in. And while you zoom in, you could have said, I might choose different numbers to see it better, but I plugged in actually rather realistic numbers. It also highlights the problem. It's called the pre-wetting phase transition, and it took like 20 years to detect it experimentally after it was predicted by Kahn and Moldovas and others. And this is because it's very close for most systems that we know, in particular polymeric systems, very close to the binodal line. So you could not really distinguish whether something happened to the surface or already have a big macroscopic condensate sitting on top. But actually, Bonn and co-workers, they managed this, so it was very exciting in the 90s. But when you plot the real numbers, this is simply what you get here as well. So it seems like not very exciting for biology because it's very unlikely that a biological system tunes this fine range. However, this is, of course, a very crude picture of the system, a binary system, so really too minimalistic. Today, I would like to convince you that when you extend this only a little bit, and simply by the ability of proteins to bind to a surface, which is kind of a common idea that proteins can do this, you can actually can strongly alter the pre-wetting phase transition, but also get phenomena like surface phase separation. Okay, and in the second part, I use this model a little bit extended to also map out in vitro data that we worked out together with the Honigmann lab in Dresden. Sorry for always moving back and forth. I don't know why it's uncharged. Good. So what's the model about? The most minimalistic model, when you think about such a system, could be still binary. So we still could think about a two-component system in the bulk and one in the membrane. But you should have at least two states. So one, the protein is in the bulk, or it is bound to the surface. So this is like really minimalistic. We'll extend a little bit later. But let's start simple. And then when you come from a large coarse-grained scale, you would write down the following kinetic equations for that system. You would write down, first of all, a Canhila type of equation for the bulk, and here also one for the membrane. So M is always the membrane. This is this surface here. And of course, because you're binding, you will have actually a singular source term for molecules being able to bind. Then you can work out the classical non-equilibrium thermodynamics, and you see like there are chemical potentials, the mu's that drive the fluxes here, and you can derive them from some free energy. And moreover, you have a binding flux, S, that's here this sink and source term. You can separate it if you want between a part that can relax to thermal equilibrium and one that can't. This is exactly that makes the binding active. Today I will focus a bit less on this, but if you want to talk with me about this, also with Frank, about this active wetting stuff, I'm happy, I'm here, I'm around and happy to discuss. Okay, so how does such an equilibrium binding flux look like? There's some subtle aspects how you choose the rates, but in principle you can choose it like this, that in the end you make sure that when the chemical potentials are equal between the chemical potential in the bulk and the bound stuff, when they're equal, then the flux vanishes. Then you're consistent with equilibrium thermodynamics. You have obviously these two equilibria in these systems, the binding equilibrium, so evaluated at this surface. Yeah, the chemical potential are equal, as I just said, but also you can have phase equilibrium, then they're just equal in space. Yeah? And this is kind of the system. Of course, these two equilibria will be very important because they will govern the behavior of the system. Free energy-wise, I don't want to get too much into discussion whether you use Huggins or something else. For the stuff I showed today, it's almost irrelevant. However, the basic structure of such a free energy should at least, I think, contain a bulk term with a density and some penalty for having a gradient, or also a surface term with a surface density, penalty for the gradient, and very importantly, a coupling. Because when a molecule is close to a surface, it can see the local energetics of the bound components, and they should be able to interact. So you can think about expanding these two fields in all kinds of orders and coming up with cross-coupling interaction parameters, very similar to a classical expansion of up to a second order Weyl coefficient. Okay, of course, you need bounding conditions. I forgot the bounding conditions where it looks a little bit like the bulk doesn't see the surface. Obviously, it does when a molecule binds. You have to make sure that when it binds, so that there's a flux normal to the surface, so when molecules come in, that actually leads to a binding event. Okay? And the last very important condition is, of course, the so-called wetting boundary condition that comes from calculating the derivative. It kind of slaves the gradient of the concentration field with this derivative of this coupling. Yeah? So whatever you put in the coupling, now you have a certain possibility to tune wetting. And for the people not so much familiar with this, that's exactly where the contact angle comes in on the left-hand side. Okay, good. This is the theory, and then you can look at this, what happens at equilibrium, first of all. And obviously now we have stuff in the membrane and stuff adjacent to the membrane, so you can come up with two order parameters in the problem. Actually, not come up, that's very important. Thermodynamics dictates these two order parameters, so there's no choice. Yeah? So if you look at what is the conjugate to the chemical potential of the membrane, it's actually simply the amount of bound molecules you have on that surface. Yeah? And the conjugate to the chemical potential to the bulk chemical potential is what is called the surface excess concentration. It's a very beautiful quantity. So exactly what you calculate is you calculate the amount of stuff here. It's not really clear, but like there's a profile. You count the excess concentration respect to the infinity concentration. Then integrate over space. So this gives you a number, but the number has now the dimension of molecules per surface area. It's a beautiful quantity, an intuitive one, and that's a conjugate one. So let's look at this. What you get. Yeah, this is a bit annoying, sorry for that. Hope we get another 2 minutes for clicking 10 times each. So what you get actually is this kind of behavior. So you can plot now the volume fraction here, starting from the surface of the membrane into the bulk. You see it kind of decays. This is not zero, it's just small. So you see, you note, oh wow, the surface is able to recruit significantly a huge amount of molecules very adjacent to the membranes. And I show here two curves, and I'll tell you in a second why. Then you can look also into the membrane, and I will call those thin and thick layer for a second. I'll tell you in a second why, and you can first of all look into the membrane. And when you look into the membrane, you see actually that the concentration of the molecules bound makes a jump as you go from very close to the membrane into the membrane. The reason is, of course, because locally the chemical potential equilibrates, and thus you have a concentration jump of the respective order parameters. So, how have we created these kind of two cases? I have looked at a case where I fixed the chemical potential far away from the surface, and looked at two different values, and the two different values are actually on the left, on the right-hand side of a phase transition. Yeah? And you see the phase transition very nicely in this Gibbs phase loop. Yeah, that shows here that there's a kink, which shows in the end it's a discontinuous phase transition. What does the order parameter do? That's quite interesting. Both order parameters jump. Yeah? And the reason why they generically always jump is because both phenomena are coupled. Yeah? So usually, when you look into the literature, this order parameter you would associate with pre-wetting. So the pre-wetting order parameter obviously jumps. Yeah? And this order parameter you would associate with surface phase separation. But now, since when molecules are close by, since they talk to each other energetically and through binding, it's very difficult to get rid of one jump and only have one other. Yeah? So which means distinguishing both phase transitions is per se not so easy. Yeah? And I will show you in a second how one can do this maybe in the near future. Yeah? But because they're generically coupled. Okay, but then what happens to this graph? Well, this was what I do now from this graph. This looks almost boring. The pre-wetting zone is very thin. I just added binding. I didn't do anything else. Okay. And that's what you see, that this pre-wetting line has broadened, and this is a logarithmic plot. So it became really now a broad regime, so it becomes now easily accessible. Yeah? And I didn't tune any further parameter. But of course, I mentioned the coupling. So the coupling between molecules being close to membranes and bound, right? And now, of course, you can get crazy because you get all kinds of combinations of the coupling. And why is this relevant? Because when molecules bind to a surface, they expose a certain other part of the molecule, and then the interaction can be different. So it can be different that they get more adhesive to their own bulk copies, so to say, or they get even less adhesive and repellent. We see this. I mean, a good example is PAG. I mean, PAG phase separates, and once they adhere, because of entropic interactions, they repel even the PAG droplet. They're also a good example. Yeah? So that really matters. I can't walk you through all these phase diagrams, but what you would expect is that each combinatoric combination of thin, thick, to dilute and dense, yeah, can actually occur. So you can have actually always four phases. So the diagrams become, first of all, complex, yeah, but the regime is first of all here broad, and you can look into these different regimes. And if you want to look up different combinations, which might correspond to certain IDPs, then please have a look at that paper. Okay, so this is on thermodynamics, but there's a problem, and because there's no coexistence of state except at the transition line. So when you go from here to here, you switch from phase 1 to phase 2, but in each domain, the membrane is homogeneous. And this is not what people see usually in experiments. There's something like this. This is a little bit of spoiler of the experiment now. They see patterned surfaces. So obviously of a dense and a dilute phase. So something seems to be missing here, so it's maybe too minimalistic. And it could be a finite size effect, but I don't believe that. I think what is missing, actually, is you're missing a component. And let me quickly show this to you. So this was actually motivated in a collaboration with Alf Honigmann in Dresden. And it's on the tight junction, where actually two cells come together and build the tight junction to create really tight connections between the cellular domains. And the current picture is very simple. So the current picture is that a receptor oligomerizes and attracts a protein, set of 1. And this protein is able to enrich such that it forms little condensates and able to recruit downstream further tight junction components, such as actin, and this actin then polymerizes and forms a full tight junction. So that's the current picture, so that in the end, phase separation of surface leads to an assembly hierarchy. And for that, let me quickly show you how that looks like in a real system. So these are two channels. This is F-actin, and this is the set of 1 channel. So it looks really like this very nicely organized tissue. And once you add latroncolin, which binds to the actin monomers, thus in the end actin disassembles, you see this nice formation of these little droplets here. Once you bleach them, you see that they're liquid-like drops. Ideally, we would like to see the assembly of the tight junction, but that happens very, very quickly, so that's very difficult. So this, motivated by these experiments and many more, we came up with the following picture, that in the end, you have this condensate formation first, then that recruits actin, and that polymerizes and forms the mature tight junction. About this one, I can highly recommend to have a look at this paper here. To finish my talk, I would like to focus quickly on this regime, how you can understand this with this simple biophysical model I have shown to you. And the model is very simple. In the end, what we add, in addition, we add a third component. So we distinguish the receptor, the protein bound to the receptor with a free lipid. Kind of allow the receptor not being bound to protein. So we have +1 component. This will actually be the crucial step. They reconstituted the proteins. So the lipids here, actually with a nickel tag, with a his tag, sorry, they are bound to the corresponding receptor that has also been reconstituted, claudin 2. And this is how they performed the experiments. And in the model, of course, you have to think about a binding process between alpha proteins, beta receptors, building these protein receptor complexes. And of course, to make volume conservation correct, you have to somehow release some water from this water layer adjacent to the membrane. Okay, here we go. What do we get? In the model, of course, we have 2 conservation laws, and one is the total receptor amount is conserved, and you come up with, when you take a binding perspective, with the occupied binding fraction in the system, which is in the end the total amount bound to the total amount of receptor in the system. And this is a fraction between 0 and 1. And when you allow the proteins bound to interact weakly, but not strong enough that they can phase separate, then the occupied binding fraction looks like this. So it goes between 0 and 1, and then here you can increase the protein binding fraction. And then there are multiple regimes. One is the dilute regime. Dilute means, in the end, the occupied binding fraction exactly scales with this one, so it doesn't care. So when you're very dilute, so you hardly add any receptor, whether it's 10 to the minus 6, 10 to the minus 4, 10 to the minus 5, you always get the same binding curve. But when you increase now the receptor amount, let's say to almost all lipids carry a receptor, you see you can dissect very nicely the effects of being non-dilute. And you would expect this because as you add more protein, you have even the transition to bulk phase separation, since systems become non-dilute. So it's in the end a measure of non-dilute binding. But if you add a little bit of stronger interactions between the proteins that are bound, make a bit more adhesive, you see that there's a new regime coming in, that is the surface phase separation regime. The surface gets kind of patterned, and below the saturation regime. Yeah? And you can summarize this nicely into a kind of a phase diagram where we plot here the total amount of receptor, here the total amount of protein, And here, when you go from a dilute to a non-dilute binding regime, you allow the surface phase separation to happen below saturation. It is a little regime because we just put some numbers, so let's put some numbers that fit this experiment. And the experiment looked like this. So you can either fix the total protein concentration and vary the receptor. What you see nicely in the experiment in 2 channels, this is the receptor, this is the protein. It gets nicely patterned, and again, phase separation breaks down, simply because of entropic reasons. The experiments have been done by Daxiao, so it's very important to highlight. And you can also fix the receptor concentration. You see a similar trend, but when you increase the protein concentration, but at a very large concentration, you see actually bulk droplets happening, and then they sit on top of these surface condensates. But you have to go to a very, very large concentration, which is far beyond physiological, but we want to try it out to make exactly the point. This is the interesting physiological regime, but at some point, as maybe most proteins do, they phase separate. But it's, of course, not very physiological. So let's in particular look at then this interesting regime, and let's see whether the model actually describes the data. And so that's exactly the same data, but just the images are just analyzed. So we calculate the total amount of protein density as a function of receptor, and here the same as a function of the protein. And you can see here, this is the average, dotted this experiment, and this is the respective concentration interfaces. There's a reasonable fit agreement. Okay, wait, it's even not a fit. So what we did actually is we extracted from the very, very low concentrated case the dilute binding affinity, and then we are left with one interaction parameter. This is just the interaction between the bound protein and the bound protein. So this is not a fit. Actually, we just played a little bit with one number, and we took the best one. You can make it a bit better agreement, but then just like we have one more parameter, I felt like it's better to show like that one parameter does the job, and the rest is fine-tuning, and I'm not convinced that it needs another lipid-protein interaction for the principle. So just one parameter. Okay, this is it, and then of course you can use it to extrapolate the experiment if you want. That's the data that we had, and you see actually there's a huge range where the system can very robustly undergo surface phase separation, and that's our current picture, that this is very relevant for the tight junction and maybe for many other membrane binding proteins. Good, let me quickly summarize. So I have 2 points to make. I showed you a more theoretical perspective here, that pre-wetting and surface phase separation are generically coupled. And then I showed you here that this theory, at least when you add maybe more component on the membrane, can also quantitatively describe in vitro reconstituted systems relevant for the tight junction. And yeah, I would like to thank particularly my collaborators, Alf, Daxiao, also Frank for the collaboration on these topics, Giacomo, Susanne, in particular, Shu Ping, who was involved in all these kind of things, my group, and you for listening. I'm sorry for the clicking today. Yeah, thanks. Better don't touch again.
Great talk, Chris. Thanks. Did I understand correctly that in the second part, the CO1 clusters are just forming by surface phase separations within the membrane?
Okay.
If that's correct, then what's the role of the bulk?
Second,
what's the role of the bulk?
Oh, okay. So what we checked, what Alf checked using AFM, it's consistent with the monodata, first of all. So this is the discussion whether it's pre-wetting or surface phase separation is a bit like maybe not that relevant, first of all. And that's why we can of course compute, we have in the theory the pre-wetting aspect as well, but even we didn't we calculated the stuff, but it's really consistent with the monolayer, so we could discard it. The bulk has exactly the role of, I shouldn't say reservoir, because there's of course a conservation law of the proteins in the bulk and the surface, yeah, but that's exactly its role. So essentially it's almost like a reservoir, so if you can really as an experimentalist throw more or less molecules in the box, because you don't lose much proteins usually to the surface, yeah, so in terms of the total number of molecules, yeah, so you can actually control that. That's the role of the bulk, control parameter.
If I may, what's controlling the total fracture volume of these droplets in the surface? Why is not the entire surface filled with droplets?
Exactly. That's Thank you very much. Yeah, but that's the idea of course of phase separation, as Sam said. The point is, so of course, if you do not have the third component in the membrane, then you don't get this. Then you have to get it just at a point. This was a little bit my pitch of saying, this may be just a binary model for the membrane is a bit too simplified. But I should note that, of course, the binary model you get for very low KD values. In the end, all proteins bind to all receptors, which means you don't hardly find a free receptor. So, and this you can actually engineer. You saw even this limit, but it was too limiting to understand the data. Exactly. But otherwise, it's the same as for classical phase separation. That there's a fixed condensed volume compared to the entire. In that case, it's a surface.
So another question over here.
In the experimental images of the Z01, I saw some domains. I don't know if they're exactly like phase separate, like circular domains, or because they look kind of like percolated. I don't know if it's related to any like heterogeneity in the receptors, or is it a phase separation?
I mean, first of all, for me, the word phase separation is there's a phase, there's another phase. Whether it's a liquid phase or relaxes on the time scale of interest or grows differently, but first of all, a phase grows because you pick up some material from the other phase. And whether the describing it with concepts from liquid-liquid phase separation that can be discussed, but when we bleach it, actually we see that it recovers, but it recovers very slowly, I have to say that. Yeah? So, and that's why, of course, in the end, in a nutshell, the surface tension is very, very low. And then, of course, when this is very, very low, of course, condensates can grow a little. They don't have to grow uniform and spherical all the time. Yeah? So then they can go like this as well. Yeah? So I think that's my answer to this.
Okay.
So can you comment on the structure of the surface bulk interaction function that you used, and sort of how to think about, as the surface binding changes, how that affects the contact angle? Do you need it to be more wettable as you increase binding in order to get this shifted pre-wetting transition to lower concentrations?
This you don't need. It's not a prerequisite. So to really get a significant shift, you don't need the function to be very, very special. So even a linear classical Duchenne choice does the job. But to get very interesting phase diagrams, where you could even see surface phase separation below the critical point of the bulk phase separation, for example, you have to think about some sort of feedback. Like, for example, as you bind, you become effectively more adhesive or less adhesive. Yeah? And mathematically, how we did this, I mean, I think I just believe in the principle, we just did a Taylor expansion up to second order, and account for all couplings between the field phi and phi m. Then you can count how many cross-coupling parameters you can vary. That's what we did.
Okay, thank you.
I was curious, in the beginning, at least in the first model, it seemed like you could almost extrapolate this to any surface, but you also talked in the beginning of your introduction about how membranes are kind of unique in their fluidity. So I could imagine other biological surfaces, like a cytoskeletal filament or even DNA, and like in which ways would all surfaces be the same, and in which ways would a sort of rapidly diffusing two-dimensional surface like a membrane be different?
I think for me, since I did not do much multi-component membrane physics here, the key thing is it's a fluid and there's diffusion. And what I also learned in this collaboration a lot, the diffusivity inside a membrane of proteins, even macromolecules being bound to a receptor, is huge. We talk about micrometers per second. Order can be lower, but I mean, this was a bit shocking. I felt like they're much, much slower. So they move very, very quick. So I think, and that's definitely a key feature membrane has, in addition to the binding. But there's much, much more. I mean, you heard about the mechanics. I mean, I think that's also relevant, but as you saw my sketches, for us, it's a support lipid bilayer that is flat, so there's no elastic effects. So yes, I think it's more general. I hope. That's a little bit what I tried to propose.
Does what you said, though, about the fast motion, could that help these, in the experiment, could that help these domains in the surface phase separation coarsen, and have they measured if and how much they coarsen?
We did. It's slow, and it's so slow that at some point other effects kick in. So we looked at this for an order of a day, and then at some point also Alf told me like, be careful, then the assay has some problem with this or that. So we stopped this because then other things happen. I think there's, I would expect that also two-dimensional protein condensates can undergo something like aging or later effect. This I would expect in general. Ripening is very interesting because you have two modes of ripening. Of course, you can think about, okay, it's a two-dimensional system, so you can eat up a smaller droplet in 2D through taking out the molecules and it moves into, very unlikely. It's much more likely that it unbinds into the bulk and goes there. It's much, much faster. So this is what I expect. But this we played a little bit, but more theoretically, not in the experiment. At least it's liquid enough that it's not a kinetic effect. But I have to say it's not super liquid. Let's put it like this. So I think they're very slowly, and you see this also in this not roundish morphology. Let's put it like this. I shouldn't say super liquid. Okay, so, yeah.
Right, I'm afraid we're out of time for this session, but I'm sure Chris will be around, so if any questions come up. So let's join the speakers of this session again.